Revati, Kirti, Veda. And usually, all let me know. Zadi, welcome. Chat and tell me whether my voice is audible, clear. Any obstacles? Okay, Revati, come on, tell me whether my voice is audible. No obstacles. Okay, welcome, Sneha. Yeah, replied audible. What about rest of the participants? Shubha, Suma again replied no. What is no? My voice is not clear? Hey, come on. Chat and tell me whether any problem in the network. Voice is clear? Shall we start the session? Still end the session. We wait for seconds. Vidya, to hear me. So, Gangotri, you sit in a place. Okay. Let me not waste the time. We'll start the session now. Those who have a problem with the network, please be seated in a place where there is a clear network so that you can have the clear voice and also clear images or slides. Okay, I'll start the session. I'll share the screen so that afterwards I cannot view your charts. If you have any clarifications, you just tell me now so that I can have that in my mind and proceed the session. Anything to be clarified? Okay, the voice is clear and I hope there is no clarifications to be cleared which were discussed in the previous sessions. Okay, I'll share the screen. Let me see. And somewhere no marking at all, plain. organism. Think this is an organism. Either it is a plant or animal. Anything. Either it is a plant or animal. The first image, there is no any markings, only purple color indication. This is an organism which undergo reproduction, and as a result it divides into two daughter cells. You observe two daughter cells. In the first image you find red mark and two variations. Each new individual organism has got variation, and that variation is not similar with the second one. The two organisms has got two different kinds of variation. One is red marking, other one is green marking. And again these daughter cells, when it get matured, they undergo cell division and they form the daughter cells are new organisms. In two the division, it forms entirely different organism with a instead of purple. What does this indicates? This indicates there is abundant of variations occur in the nature. Abundant of variations. Do you think that these variations helps the organism to survive in this environment? Yes. Maximum organisms, they survive because the variations is as per requirement many times, but the organisms may not survive because of more variations, unexpected variations, not clearly found in the new organism. And what are the reasons to bring up these variations? Yes, the changes in genes, definitely yes, there will be changes in the genes. For each and every trait, that means character, genes are responsible. For each and every trait, one particular gene is responsible. And these genes decides the characteristics in an organism. If you find red band or green band or blue body, definitely there might be changes in a genetical level, or in find the variation among the new organisms. What is this total process called? Are the springs, or for the next generation. Offspring in the sense next generation. And this process is known as heredity. So heredity is very essential, where the genes get transferred from parents to their offsprings, and that decides the traits or characteristics among the organisms. Always the characteristics go on changing from generation to generation as we find change in the environment. In the same way, you find changes in the organisms also. The organisms which undergo changes, which can easily adapt to the environment, will survive, whereas rest of the organisms will die. So many of you all watched the movie Jurassic Park. When these dinosaurs are found, mainly dinosaurs or dinosaurs, completely and very new organisms came on the earth. In the same way, the human beings came on the earth as we can adjust to this environment. And when the organisms get adjusted to the environment, when these organisms attains maturity, when they undergo reproduction, they gives rise to young ones. During that process, if the parents are very tall, the children are also very tall. If the parents are very dwarf, the children too very dwarf. What all these indicates? The character like tall, the character like dwarf is representation of a gene, and these genes passes from parents to their offsprings, and hence if the parents are tall, even the children will be tall. If the parents are dwarf, even the children will be dwarf. In the same way, you find the things in the plants. When the tall plants produces, if they produce the seeds, the plants will be dwarf. What this indicates? Characteristics offspring, and this process is known as heredity. Yes, in this session we are going to know more about heredity. You see the diagram. Our human beings have earlobes, which is completely attached in B and free in the diagram A. What this indicates? What does this indicates? Okay, the earlobes is not common to all human beings. Few human beings have free lobes and few human beings have attached lobes. This is because the genes may be dominated or recessive. So, the reason behind these earlobes are the genes may be dominating genes or recessive. If the genes are dominating, they can have the free lobes. If it is recessive, they have the attached lobe. In the same way, we can find the dominating factors. If any gene that is recessive, that will be suppressed. The gene will be found, but it is not expressed. It is suppressed. And not suppressed. Okay. As you see in the second row, the tall plant into tall plant, And that tall plants gives, how come the parents are too tall? The one ratio of the small. So for that, you have to see F1 generation, the first row. In the first row, tall plant and the dwarf plant crossed. How the crossing takes place? Think the pollen from short plant and ovary from the long plant is crossed. That means cross pollination. Cross pollination is done. Then you see that F1 generation is having only tall plants. This concept is little bit difficult to understand, but there is no other way to understand in a simple way. Now I'll repeat. Crossed with short plants will be G is the tall. What is the letters indicated? Capital T, capital T. One capital T comes from the tall, one small t, small, and hence plants are capital T. The factor for shortness or dwarfness is suppressed. Suppressed in the sense the gene is there, but it is not expressed. So, capital T, small t when you find in heterozygous condition. Heterozygous means one capital T, one small t. That is one gene for tallness, one gene for dwarfness. Both are present. Then you call heterozygous tall plants. Then what will be the ratio of one? Only one. That means all are tall is the ratio. One. Okay. In the same way, you just watch the second one. When F1 generation seeds are selfed in the sense capital T, small t. Look at second row. Another one, capital T, small t. They are selfed. F1 generation seeds are selfed. Then you find capital T, capital T, capital T, small t in 2, small t in 1. That means capital T anyhow it is tall plants. Capital T, small t is also tall plants. Small t is short plant. Here, when the recessive genes come together, that is small t, when it is come together, there is no dominating factor, then that plant will be short. Only there you can find the short plants. So this is the analysis. This is the explanation given by the father of genetics and his Mendel. Mendel explained this with the help of checkerboard. Mendel was educated in the monastery and went to the study science and maths in the University of Vienna, and because of failure in the examinations he didn't get teaching certificate, so he went back to the monastery and started growing some vegetables in the kitchen garden, and there he come across pea plants, very easily grown. Within couple of months he'll get the yield, and that seeds again he can regrow. So he selected the pea plants for his further research. There he started the work, and he selected the pea plants because of very interesting factors, and those interesting factors are why the Mendel selected pea plant for his studies. One, the character is the seed shape. He found two shapes, that is round and wrinkled. And constricted or inflated, and even pod color, that is green or yellow. He selected the other feature. Before forming the seeds, they produce flowers, and the flowers are purple and white. And he found two sizes of the plant, one is tall and one is dwarf. And even he found one variation of existence of the flowers, that is axial or terminal. Axial or terminal. So these are the 7 contrasting characters Mendel found in the pea plant. Let me see with neat diagrams. So this is pea. The second one, seed is wrinkled. Seed constricted. Flower location, axial, terminal. You observe keenly where is the axial position and where is the terminal position. Terminal, only one at the end. Axial, you find it sides. Plant size, tall and dwarf. These are the 7 contrasting characters Mendel found in the pea plant. So Mendel selected pea plant for his experimental study. This is very important concept. Important. And the pea plants are so easy to grow. In any climatic conditions we can grow the pea plants, either on the ground, pot, anywhere you can grow the pea plants, and the lifespan is so short. Within couple of months the life cycle will complete. It produces seeds, it provides yield, it will die, and again very soon, within a year, you can have more generations. So he selected the pea plant. It is hardy, it can adjust to any environment. Lifespan is contrasting characteristics, so many contrasting characteristics. So he can study in a different combinations. So he selected the pea plant as easily it undergo cross pollination. Easily it undergo cross pollination so that he can study the variations. When there is a cross pollination, he can have the chance to study more variations. Hence, he selected the pea plant for his experimental study. Now, you're seeing this diagram, the picture where the picture explains monohybrid cross. Who gave this monohybrid cross? Again, Mendel. In his experimental studies, he went with the numerical of mathematical calculations combinations. He went with tabular, and those tabular columns are called checkerboards. The tabular columns are called checkerboards. What you are seeing in the box, both are small t's. Select one small t and you cross it, you will get F1 generation, that is capital T small t. What is the ratio of F1 generation? 1. 1 is the ratio. And you see in the second F2 generation, how to write checkerboard? Put in a box. Capital T small t, 9. Orange and green boxes and yellow boxes, you just keenly observe that. Now you multiply 2 orange boxes. Second boxes. Capital T. And now you multiply 1 orange box with the green. Capital T small t. Just write it in first line. And you go for second line now. Small t, capital T. But we represent all writing first, recessive second. Even though while writing you should write small t and capital T, but usually we write dominating first and recessive second. Capital T small t, and now green and green, small t. This is about F2 generation. Now tell me, what were the output of the plants? Will be the size of the plant? Yes, it will be tall. Second one, capital T small t. How many are there? 2 in number. Those 2 are also tall. Even in the heterozygous, dominating factor is found. It results in dominating character, means tall plants. And look at the fourth one, small t. That is dwarf. Genotypic. Genotypic ratio, different kinds of genes. Which one the different kinds of genes? Capital T, small t. These are the 3 combinations of gene combinations. So 1:2, 2:1. genotypic ratio in the monohybrid cross. 1:2:1 genotypic ratio. Look at the second one, phenotypic ratio. Phenotypic means how the plants appear, external morphology. Phenotypic, external morphology. When you see the diagram, 3 are tall, 1 is small. Same way you put in a ratio, 3:1. You're not counting the genes, you're seeing the plant and writing the output. 3 plants are tall, 1 plant is dwarf. So, the ratio is 3:1 ratio. So, this is about monohybrid cross. The cross between 2 plants with one different contrasting character is known as monohybrid cross. The cross between 2 different plants with one contrasting character is known as monohybrid cross. Okay, my voice is Debates of Life channel is wishing all of you as have. Okay, instead of all participants, wish her very warm good morning. Okay, before entering into the second session, that is dihybrid cross, let me know that anything's to be clarified here. Come on, chat and tell me. Anything should be cleared? Okay, voice is audible, okay. Whether the concepts are okay? Anything should be repeated now? Any concept should be repeated now? Come on. We discussed about 7 contrasting characters of the pea plant and why the pea plant is selected by the Mendel. The Mendel came back to the monastery. Instead of teacher, he became a gardener and he was a very good mathematician. So, his mathematics principles were not understood clearly when many scholars watch his research observations. Many they didn't understood what he wrote. Such a numerous of permutations, combinations, calculations he put in his observation record. Only after his death, his work been identified and he was awarded as father of genetics. Anything to be clarified? Shall we start with the dihybrid cross? Chat. Venkata Raju is saying that it should continue with the video call because once if I go with the video call, the camera will be on and I cannot share the screen. So, For explaining this, okay, we'll go with the other session. That is dihybrid cross. Okay, this is the image explaining about dihybrid cross. Two kinds of seeds are selected. One seed is yellow and round. The other one is green and wrinkle. The gametes for that is for yellow and round. Y, R, put in all capitals. For green and wrinkle, small y, small r. This is green and wrinkle seed. And you observe keenly, both the factors should present because this is dihybrid cross. Both the factors means for yellow and round, for green and wrinkle. Both characters should found in the gametes. From and in the F1 generation, green and wrinkle, the gametes are small y, small r. When these are crossed in the F1 generation, what will be the output? Both capitals and smalls. Capital Y, capital R, small r. Same how you found in monohybrid cross, same thing. But here two characteristics are taken. There is only one character, tall and short. Here yellow and round, green and wrinkle. Two characteristics are small y, capital R, small r been selected. And obviously when dominating and recessive factors are found, which will be expressed? Yes, dominating characters will be expressed. Even the recessive are found, they don't express, they will be suppressed. Only capital letters, dominating characters will be expressed. So, what is the output of F1 generation? Now tell me. All plants are, if it is capital Y, yellow. If it is capital R, round. All plants are yellow and round seed bearing plants. Yellow and round seed bearing plants. F1 generation. What is the ratio of F1 generation? 1. 1 is the ratio because all plants are yellow and round. Ratio is only 1. Now, F2 generation is very complicated. Now, how to take the gametes from F1 generation? I said while taking the gametes, you should have all the characters. Now, F1 generation is, output is capital Y, small y, capital R, small r. You write it in one place and try to separate the gametes. The gametes are, first one, you see at the outside the checkerboard what are written in the vertical and horizontal line, those are the gametes. Capital Y, capital R, this is one set of gamete. Capital Y, small r, this is other set. Small y, capital R, this is another set. Small y, small r, all four combinations been finished. Now same thing you write in vertical column. What you wrote in the horizontal line, same thing you write in a vertical column. Same thing, you just observe now. What is given in the horizontal line, it is same with the vertical column. Observe it clearly. Okay, Revati has got doubt. Ma'am, please explain me once again about generations. Beautiful garden, Vitika. Hi, ma'am. Very warm welcome to my channel. Actually, the session is related very professional about genetical concepts. Thanks for your entry. And before exit, please give me a thumbs up. Okay, Revati, you have what is generation or genetics? What is to be clarified? What is generation or what is genetics? Generations is very simple. The parents are belonging to one generation, that is F1 generation, filial generation, and their children comes under F2 generation. Remember, parents comes under F1 generation, first filial generation. Their children comes under second generation, second filial generations. Okay, you have doubt with the genetics, right? Genetics is a process where the characteristics are transferred from parents to their children or to their offsprings through the genes. Should I repeat once again? Genetics is a process where the traits are transferred from one generation to the another generation. That is from parents' generation to the offspring. Okay, shall I share the screen? Any other concepts to be clarified? Anything to be clarified? What about rest of the participants? So voice is audible. What about Gangotri? She had problem with the network. Now, was that cleared? Gangotri? Okay, Gangotri is wishing me good afternoon. Good afternoon, everyone. Now, I think should present one side capital and small, the other side capital and small, other side only small. All the combinations you should pick up and write in horizontal and vertical line. Then multiply it. What is first line? Capital R, small r. Third one, capital Y, small y, capital R, small r. And the fourth one is capital Y, small y, capital R, small r. Now, what will be the output of these 4 plants? Yes, all 4 plants are yellow and round, because in each box there is one capital Y, one capital R, so it will be yellow and round seeds. Okay, now look at the first column. Capital Y, capital Y, capital R, first one. Second one, capital Y, capital R, small r. Third one, capital Y, small y, capital R. Fourth one, capital Y, small y, capital R. Okay, what will be the output of these 4 seeds? Yes, they are also round and yellow. Yellow and round. Why? Because in every now you see 1, 2, 3, 4, 5. 5 yellow seeds there, right? The combinations capital Y, small y, small r. And you just observe the shape of the seed. The seed is wrinkled. And you observe the second one. Capital Y, small r. How is the shape of the seed? Wrinkled. Now come to the down, yellow one. Capital Y, small r, wrinkled. So these 3 are round and wrinkled. 3 are yellow and wrinkled. Okay. Round and yellow seeds are 9 in number. Count in the picture. The first horizontal line, the first column, and when you join this you will get triangle, and those 2. Now put one triangle there, you will get 9 round and yellow seeds. 9 round and yellow seeds. And just now I said small r's, 3 small r's, yellow and wrinkle seeds, 3 number. Now come to the green one. Small y, small y, small y, small y, small y, small y. They are green. And capital R is there, round. Green and round. How many number are there? 3 number. And the last one. All are small letters. Small y, small r. That is green and wrinkled. Now look at the ratio. Yellow and round 9. Yellow and wrinkle 3. Green and round 3. Green and wrinkle 1. This is known as dihybrid ratio. 9:3:3:1 ratio. 9:3:3:1 ratio. This about dihybrid cross. Okay. All of you come to know what is dihybrid cross? Anything to be clarified in the dihybrid cross? Come on, chat and tell me. Dihybrid cross is cleared? Okay, Gangotri replied as cleared. She understood, I think. What about others? Okay. I should repeat monohybrid cross. Okay, I'll go for that. Okay, look at this picture. Here only one character has been selected. That is size of the plant, tall and dwarf. For tallness, the gametes are capital T, capital T. For dwarfness, small t, small t. When you take the gametes, only one letter will be selected here because it is monohybrid, only one. From tall, you will separate the gamete, capital T. From the dwarf, you separate the letter small t. Put it one side, capital T, small t. And that will be the F1 generation. If it is a capital letter, what will be the and in the F2 generation, write the gametes, that is capital T, small t in horizontal line. In the first line, capital T, and the other one capital T, small t. In the second line, capital T, small t. The other one is small t, small t. So, 3 plants will be tall, 1 plant will be dwarf. So, phenotypic ratio, that is how we can see the plants. Appearance, morphology. 3 are tall, 1 is short. So, What is the ratio of phenotypic ratio? External appearance 3:1. And what will be the genotypic ratio? 1:2:1. 1:2:1. So genotypic ratio is 1:2:1. Phenotypic ratio is 3:1. This about monohybrid, and we also discussed about dihybrid cross. Now let me tell how the plants will grow tall and how the plants will grow short. Very clearly I told it is because of genes, and how the gene perform the action on the plants. Each gene stimulates to synthesize the protein to activate the enzyme. If the gene activation is good, then enzyme activity will be strong and hormone synthesis will be very active so that the plant will grow tall. Just now I told genes are very responsible in deciding the character, either it is tall or dwarf. Enzymes. If enzymes are good enough, the hormone synthesis is also good. If hormone synthesis is good, in previous session, phytohormones, plant hormones, axin, gibberellin, cytokinin, those hormones. If the protein activates the enzyme to synthesize good amount of hormone, then the plant grows sufficiently, vigorously, gigantically. If the gene is not active enough, the hormone secretion decreases. As a result, the plant will grow stunted, very short. So like this, the genes has a major role in activating the enzymes, in turn activating the hormones, and that hormones are very essential in deciding the length of the plant, either it is tall or dwarf. So like this, the genes has a major role in deciding the characteristics of the plant, either the plant grow or. So all this will be decided by the genes term genetic, where transfer of characteristics from one generation to the another generation takes place, and beyond this, genes has a major role. Gene in the sense DNA plays a major role, and we already discussed. In order to bring up more variations, sexual reproductions plays a major role, and even I told you all that during meiosis, genetic recombination takes place. As a result, you find the variations, and variations are very essential to adapt to the environment. If not, it will become very difficult to adjust and they won't survive in the environment. So sexual reproduction results in variations, and variations are very essential to adjust to the changing environment. And variations always comes from changes in the DNA or genetical level. This never you find in asexual reproduction. If bacteria divides 1 into 2, If bacteria divides 1 into 2, it is almost Xerox copy of parent bacteria. There is no variations. And do you think that asexually reproduced cells will all survive? No. When there is no variations at all, very minute variations, in those organisms the survival rate obviously decreases. So, heredity plays a major role in bringing up variations, different combinations, as you see in this diagram. The first one, no variation, when it gives rise to two individual organisms, two different of variations. And the second generation, when it again continues for next generation, furthermore changes. All this because of sexual reproduction. Anybody having any clarifications to be done? Welcome for my channel. Thank you for your like. सब अच्छे हैं और सोच रहे हैं आप भी अच्छे हो. Revati and others, anything to be clarified now? Debates of Life is wishing Vitika as hi Vitika. So hi from Vitika. I hope she exited. Okay, there is no things to be clarified. Okay, Debates of Life. Very glad to see you in my channel. Okay. Other response, even she don't have any doubts or things to be clarified. Kirti too. Kirti don't have any doubts. Okay. What about others? Let me know who were in this session till last. What about others? They quit. Okay, Venkatraju. Venkatraju, who is the participant there? Okay, Kirti don't have any doubts. Very good, Kirti. This is you all should go through repeatedly. Who is in the name of Venkatraju? Who is the participant there? Okay, let me clarify. If there is no things to be clarified, shall Okay, Vidya. Okay, Vidya. I hope the concepts are clear for you. So we should recall the concepts like what is monohybrid ratio, dihybrid ratio, what is heredity, what is dominating factors, what are recessive factors, and the checkerboards, and how the genes instruct the plant to grow. You can watch this video again and again till the doubts can be clarified. So I'll wind up this session, and the number is not increasing. There is a class so that we can have more number of participants. Okay, thank you. We'll wind up this session.